Through their research, scientists have successfully regenerated neurons in mice that have been paralyzed due to spinal cord injuries. This medical breakthrough has resulted in the restoration of the mice's ability to walk. Achieving functional recovery is heavily reliant on directing cells to their natural target region. A research group from UCLA, Harvard University, and the Swiss Federal Institute of Technology has discovered a vital element to restore functional mobility following spinal cord injury, according to a recent study conducted on mice. The group of neuroscientists has demonstrated that the revival of specific neurons back to their original target regions is essential for recovery, whereas haphazard regrowth did not yield the same results. In the year 2018, a study was published in Nature that detailed a technique for stimulating the regrowth of axons in rodents that had suffered from spinal cord injuries. These axons, minuscule fibers that interconnect nerve cells and allow them to communicate, were able to regenerate even in cases where the spinal cord injury was severe. However, the study also found that restoring functionality remained a significant obstacle, despite the successful regeneration of the axons. The latest scientific research, which was published in Science this week, was conducted to determine if it was possible to restore functionality after spinal cord injury in mice by directing the regeneration of axons from particular subpopulations of neurons to their natural target areas. The team began by utilizing sophisticated genetic analysis to identify clusters of nerve cells that facilitate the restoration of walking capabilities following a partial spinal cord injury. After conducting research, it was discovered that the mere regeneration of axons from nerve cells across the spinal cord lesion did not have any noticeable effect on functional recovery. However, when the approach was refined to incorporate the utilization of chemical signals to direct the regeneration of these axons to their original target location in the lumbar spinal cord, a marked improvement in walking ability was noticed in a complete spinal cord injury mouse model. Michael Sofroniew, Maryland, PhD, a professor of neurobiology at the David Geffen School of Medicine at UCLA and one of the senior authors of the study, commented that their research offers valuable insights into the complexities of axon regeneration and the prerequisites for successful functional recuperation in spinal cord injury patients. He emphasized that the study underscores the importance of not only regenerating axons across lesions, but also directing them towards their intended targets for optimal neurological recovery. According to the authors, there is great potential in comprehending the reintroduction of projections from specific neuronal subgroups to their natural target areas for the advancement of therapies that aim to restore neurological functions in larger animals and humans. Nevertheless, the researchers also recognize the intricacy of promoting regeneration over more extensive distances in non-rodents. This complexity necessitates strategies that have delicate spatial and temporal features. Despite this, they ultimately conclude that the implementation of the principles presented in their work will provide the key to meaningful repair of the injured spinal cord and may expedite repair after other forms of central nervous system injury and disease. The team of researchers was comprised of scientists hailing from various institutions, including the NeuroX Institute, School of Life Sciences, Swiss Federal Institute of Technology, EPFL, the Department of Neurosurgery at Lausanne University Hospital, CHUV, and University of Lausanne, UNIL, the Center for Interventional Neurotherapies, NeuroRestore, the Wyss Center for Bio and Neuroengineering, the Department of Clinical Neuroscience at Lausanne University Hospital, CHUV, and University of Lausanne, the Departments of Bioengineering, Chemistry and Biochemistry at the University of California, Los Angeles, the Bertarelli Platform for Gene Therapy at the Swiss Federal Institute of Technology, the Brain Mind Institute, School of Life Sciences, Swiss Federal Institute of Technology, the M. Kirby Neurobiology Center, Department of Neurology at Boston Children's Hospital, Harvard Medical School, Boston, and the Department of Neurobiology at David Geffen School of Medicine, University of California, Los Angeles, the Defitech Foundation, Wings for Life, Riders for Riders, Wyss Center for Bio and Neuroengineering, Swiss National Science Foundation, particularly PZ00P3_185728 for MAA and PZ00P3_208988 for JWS, Morton Curie Paralysis Foundation, specifically for MAA, Alarm Foundation for both MAA and GC, and the Dr. Miriam and Sheldon G. Adelson Medical Foundation, granted to MVS, ZH, and TJD, were instrumental in supporting this project. Furthermore, Wings for Life provided additional funding for MAA, MVS, MAS, and MMJWS received support from the Holcim Stiftung Foundation, while JWS also received support from the Canadian Institutes for Health Research. We would like to express our gratitude to J. Ravier and M. Buri for their illustrations, as well as L. Bhatti and I. Gantar from the Advanced Light Sheet Imaging Center, ALICE, at the Weiss Center for Bio and Neuroengineering, Geneva. Additionally, this project was partially supported through the resources and services of the Gene Expression Core Facility and the Bertarelli Platform for Gene Therapy at the School of Life Sciences of EPFL. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.